Okay, hello everyone. This week we will be discussing a new type of analysis, correlation, which I'm sure all of you have heard of before. So in correlation, we are studying the relationship between two numeric variables. And here is a small example, how across mammals, across different species of mammals, body mass on the x-axis here and brain mass on the y-axis, how they are related to each other. Each dot is a species, and its both body mass and brain mass have been measured. And as you see, there are some small species which are small in both body and brain, and there are some relatively large species um, who are large in both, again, whole body and brain. And people, biologists have been studying such relationships, both to understand what kind of characters are related to each other, numeric quantitative characters are related to each other, and also what kind of deviations may we observe. And here you might already notice that, for instance, um, humans here have a relatively high brain mass uh, compared to organisms like porpoises, uh, which have much larger body masses. So, in fact, this kind of questions about deviations, so we will continue studying in the next chapter about regression. But correlation is a tool that measures, simply measures whether there's a relationship or not. And this is a very simple example in that the relationship is very obvious to the eye, but sometimes some relationships might not be so obvious. So this is relate a similar type of problem, um, again about phenotypes across species, and whether different phenotypes, different characteristics are related. And here, in the study from 1996, this guy was trying to understand whether across primates now, um, neocortex size, so part of the size of some part of the brain, some part of the frontal cortex, is related to the social group size of the same species. So both variables have been normalized to a mean of 0, as you see. So they are scaled. And each dot, again, represents a species. And here, again, the question is whether the two variables are related to each other, related, if one might be, of course, if we can conclude that they're related, that means one might be influencing another, or both might be influenced by some third factor. Um, and what we will be learning in this week's lecture is, for one thing, how to measure correlation, how to come up with a statistic that tells us the relationship, tells us about the relationship, um, and also how to calculate statistical significance, how to construct confidence intervals, how to calculate a p-value for whether such a relationship might be random or not, how much can we trust the reproducibility of such a relationship given the noise in the data, uh, and and also when assumptions of certain tests are violated, what kind of alternative correlation tests we can use. We'll also be learning about that. Okay, so first, the first topic will be a measure of correlation, and the correlation coefficient is that measure. It's a value that measures the strength and also the direction of the really linear relationship between two numeric variables. Um, so its value indicates how strong the relationship is, and also its sign, positive or negative, indicates uh, in what direction the two variables are correlated. You can compare this, the correlation coefficient, conceptually with the odds ratio, which you might remember. It's a measure of how strongly two categorical variables are in their frequencies. So here we're in the correlation coefficient, like in these kind of problems, we're talking about two numeric variables. The odds ratio was a value we calculated for two categorical variables that had two levels each, two types each. And we the odds ratio would deviate from one, uh, would become much higher than one or much lower than one, uh, depending on if the relationship was strong. And the the log transformed odds ratio, you might also remember that would was centered at zero when there was no relationship, and it would deviate towards positive values and negative values when there is a relationship. The correlation coefficient, different from the odds ratio, it is bounded by -1 and 1. We'll see better now. So just to wrap up this last point, the book has a nice summary of different type of statistical tools that are commonly used when we are studying the relationship between two variables, and depending on whether which one is the response and which one is of explanatory type, if such hierarchy, if such a relationship can be established, we use a different type of tests. So if both are categorical, as we mentioned, the test to use is contingency analysis, the chi-square or goodness of fit test, or the Fisher's exact test. If both are numerical, then we can use correlation, linear Pearson's correlation that we will learn today, and also Spearman's rank correlation, which we'll also learn today. And regression is another type of analysis that we will learn in the next week. There are also these other tests we started learning. So if the response variable is numeric and the explanatory variable is categorical, so for instance, if we have two groups and some numeric variable measured for the members of the two groups, um, so that variable itself, let's say height, is the response, and the group itself, it's also variable. It's a categorical variable, right? And so we can study the relationship, the group there would being the explanatory variable. We can study their relationship using a t-test or a Mann-Whitney test. If there are more than two groups, we can use ANOVA, etc. And then there are also these situations where the response can be categorical, can be 1 and 0, many in some cases. For instance, did a person die or did a person survive? And the explanatory variable might be numerical, like how many cigarettes a person smoked by the age of 60, and by the age of 60, did he or she die or survive? And so for these kind of problems, the tool we have available is called logistic regression. We'll briefly touch upon that in, again, in next week's chapter. Okay, So we said we need a measure, we can come up with a measure of that captures how related two numeric variables are. And what kind of measure, how could we, if I asked you to construct such a statistic, what kind of approach would you use? So somehow it has to tell us about how the togetherness, the togetherness of the two variables, the values of the two variables, uh, how much the x and y values, each point is represented on the x and y axis, right? How much these x and y values deviate together from the mean. If there was no relationship, we would expect data points all over the place, right? So they would be deviating in this direction and also this direction, and this direction equally frequently. And then we would say there's no relationship. Alternatively, the x values might be high only when the y values are also high, and x values might be low only when the y values are also low. In this type of situation, we can we can infer there's a strong relationship. So somehow we need a measure to capture this togetherness, how x and y are deviating from the average, from their averages. And the Pearson correlation coefficient, after Carl Pearson, from more than 100 years ago now, captures that behavior. So it's this formula, not very, it looks a bit long, but it's pretty simple. Here we have, for each of the values in our data set, for each point, we calculate the deviation between that point and the mean on the x-axis. So the point's value on the x-axis and the mean of the x-axis values, and for the same point, its value and on the y-axis and the y-axis mean. So basically, for so here, for instance, if the mean of both sets are 0, in this case, we would be multiplying here. We would be calculating, sorry, first the difference between this value's x-axis value and the mean. The mean is 0, so it would be something like 0.07 perhaps. And so we would write 0.07 times, again, this value on the y-axis, this would be 0.025, and its difference from the mean being 0, so 0.025. Okay, that would be the product for this point, and then we would calculate the same products for all the points in our data set. Okay, that's this sum here, going from 1 to n. Okay, and we would calculate those sums. Some of the values would be, so here the differences are both for this point, the differences are both positive. For instance, for this point, this product would have a negative value. Why? Because on the y-axis, its value is above the mean, right? But for the x-axis, its value is below the mean, right? So it would have a negative value. So some of these products will be positive, some of them will be negative. and we will sum all of these up. Okay, so that's the numerator. And the denominator, so this part, sorry, perhaps I should also mention that the sum of these products, sum of the products of differences, it will be capturing that togetherness behavior or joint behavior of the x and y values. If there's a lot of if all behavior is parallel, is correlated between x and y, then this sum will be a strong positive value. If the relationship would have been negative, then the sum would be a large negative value. If there was no relationship, if the data points had been all over the place, like if we had also points up around here as frequently, I should add more here, then the positive values and the negative values will cancel each other out. As you see on the, let me, do I have a, in this quadrant, if you see, and in this quadrant, the products, the sum of products will be positive, because two positive values or two negative values that cancel each other out. Whereas in this quadrant and in this quadrant, for these points, the products will be negative. So if we have equal numbers of points all over the place, the sum here will be 0, obviously, right? But if we have few points over here, the sum will be much larger than 0. And the last possibility is, of course, if all the data are in these quadrants, but not here, then the sum will be much lower than 0, right? Be negative. Okay, so this is what the numerator captures. I hope everybody is clear on how this is calculated. And then on the denominator, we have formulas that are familiar to us. This is just difference between each value and the mean of that set of values squared. So the sum of squares, right? Sum of squares we know from ANOVA. We also, this is also the numerator of the variance. If we divided this by n-1, it would be an estimate of the variance for x, and this is the sum of squares for y. If we again divide it by n-1, it would be the variance of y, and the square root of these values. So basically, this division by basically the square root, the square roots of the two sum of squares. It's not the standard deviation, as you see, because we haven't divided by the sample size -1. So this part, it works as a scaling factor, and it constrains by dividing by the sum of squares. We constrain this value r to lie between -1 and 1. Okay, so in the most extreme case, if x and y are totally correlated, if the values of one depend fully on the other and there's no noise whatsoever, if all the values lie on a single line, okay, there's no variance whatsoever, then the sum of the products here, I guess it should be obvious to all of you. The sum of the sum of the products of differences here in the numerator will be equal to the square root of the sum of squares multiplied. So these will be the same values, and we will have 1 as the correlation coefficient. If we have just the opposite, if we have a totally negative relationship like this, then again, these will have the same magnitude, but each of these products will be negative, right? So the smaller the x, the larger the y. The larger the x, the smaller the y. So we will have negative values, but of the same magnitude here, so they will cancel each other out. We'll have -1 as the correlation coefficient. Okay. Yeah, just a reminder. So the numerator is a value that is related to a statistic called covariance, and these are related to the variance, obviously. And yeah, the covariance basically captures the relationship between two variables. And by dividing the sum of products with the sum of squares, we end up scaling this value such that it's distributed between -1 and 1. If you can't see a correlation coefficient which has a value of 2. Okay, I mean, in with real numbers, you will always get a correlation which this formula will always give a relationship that is constrained between -1 and 1. Okay. The Pearson correlation coefficient, it is shown with the symbol R, and R is usually used for samples, and rho can be used for to describe the population correlation coefficient. So let's say we're interested in the relationship between, again, social group size and brain neocortex size across mammals, let's say, or across primates. But the sample we collect, of course, doesn't represent the full population of species of primates. And we thus the correlation coefficient we calculate is an estimate for the theoretical population correlation coefficient. Okay, but there I should add a small note here, which is that rho here, the Greek value, it's also frequently used to refer to the non-parametric correlation coefficient calculated for samples. So there's a there can be a bit of confusion there. So, but in this course, we follow the book. So rho, we will refer to rho as the population correlation coefficient. Okay, and just as we described, this is another example of two variables on the x and y axes. With we see a positive correlation, as you see, and their means are shown here, and the points, each point again represents an observation which was measured for both x and also y. And if we see if all our observations are concentrated in these two quadrants, then obviously there's a positive correlation. If they were equally occurring in the other two quadrants, then there would be no correlation. Okay, so as a small thought exercise, let's try to guess what kind of correlation coefficients we might expect in these different scatter plots between x and y values. So here, for instance, what do we expect? If we just divide, I mean, we don't have to really do this, but we can see that data are distributed across most of the quadrants. There doesn't seem to be much of a strong relationship, right? The R correlation coefficient must be pretty small. Here, though, the we seem to be observing some kind of positive relationship. How strong it is, whether this could happen by random or not, that's another question we'll address in a second. But the relationship seems to be, whatever there is, seems to be positive. And here, the relationship seems to be in the negative direction. As you see, we have a deficiency of values here and an excess of values here, these extremes. And here, this should also have a positive correlation coefficient. And if we compare with this scenario, the correlation coefficient here seems to be stronger, right? That's the main effect. And indeed, the correlation coefficients calculated from these data are apparently as follows, as shown. Yeah, so some further exercise. Let's consider these cases. What would be the correlation coefficient in these different situations? So on the top row, so here should be very high, right? Positive, very high. And then here, there's a bit of noise. So the more noise, the weaker will be the correlation coefficient. And eventually here, it's just a ball, right? The full round symmetric ball. Is there any relationship between the two variables here? No relationship, obviously. And here it's weakly negative, more negative, very strongly negative. And here there's no variation whatsoever, as you see. No variation meaning no, the behavior of one variable directly determines the behavior of the other variable. There is no deviation from that behavior, and the correlation coefficient would be very high here. Here, okay, this might be slightly tricky. So, okay, I can guess that most of you say, okay, here R is 1, but what is R here? Or here? There's again a positive relationship, but the slope is weaker. Yeah, we'll learn more about slope next week. But what would R be here? So R tells us how coupled the X values are to Y values. And here, even if the slope is weak, we see full coupling, right? An X value is, how to say, the highest X value is also the highest Y value. The second highest X value is the second highest Y value, and so on. So here, in fact, there is also a perfect relationship. So R will also be 1 here. And here R will be 0 because it's totally flat, but until that point, R will be 1. And here they will be negative, -1, -1. And how about here? So this is a particular, these cases are of particular interest because, in fact, different from this scenario, for instance, here we see a relationship between the two variables, right? Mustache relationship, some kind of up and down. Here, another case of relationship, another case, another case. So the X values and Y values, they're not just randomly related, but the relationship is, sorry, in fact, here, what's going on, we have two distributions, normal like distributions, and no relationship. But here we have this type of pattern. It can only arise if X and Y values depend on each other. X and Y values depend on each other, but their dependence is non-linear, as you see. And here it's also non-linear. And here it's also non-linear, right? So in all these cases, we have non-linear relationships between X and Y. But because the correlation coefficient, as we mentioned, it can only capture linear relationships. Okay, here the R values will all be zero. Will all be zero. Okay. Yeah. So I hope that part is clear, and if not, please ask during class and we can discuss a bit more. Okay, now we'll continue with an example from the book about sex differences in finger length. Apparently, the fourth digit, this finger here, tends to be slightly longer in males due to, turns out, testosterone exposure during development, and tends to be shorter in females. And apparently um, there is some variation among humans in the receptor that responds to testosterone in the blood, called androgen receptor. So the androgen receptor binds to testosterone, and then it has this ligand binding domain, and then it goes and binds to DNA and starts transcribing genes. That's a classical hormone receptor transcription factor. And these are called nuclear receptors because they work in the nucleus, but they also have receptor function. And apparently some variation in the sequence of this gene, which involves repeats, so a repeat of CAG within the gene that encodes for, I forgot what amino acid, glutamine perhaps. It occurs in different numbers in individuals, so from 18 to 28 in the sample, close to 2 times the difference. And the number of repeats seem to be influencing the function of this testosterone, the androgen receptor, which binds testosterone. So the people who led the study, they measured the number of repeats in different people's 46 men, in across 46 men, the number of CAG repeats in their androgen receptor, and their second digit to fourth digit ratio. And this is on the first column here is the number of repeats. The second column is the ratio. And the lower the ratio than 1, although it's sometimes it can have values apparently higher than 1. The lower it is from 1, the higher the fourth digit length is. And these researchers want to study if there is a significant effect of the number of repeats shown on the x-axis, because it's potentially explanatory, the number of repeats, and the 2D to 4D ratio. Yeah, if there's a relationship, it looks as if the higher the number of repeats, the higher the ratio. So perhaps the protein becomes, well, you can think about it, how the number of repeats, how it might be influencing the function of the protein. But that's a biology question. Okay, the question we have for the biometry class, the question we'll try to answer is whether the relationship is reproducible or not, and how strong the relationship is. Looking at the values, there seems to be some pattern, right? So but there's also a lot of noise, so it's not easy to say uh, how that this is a definitely reproducible pattern. Whatever we will conclude in any case, beyond describing the data in a graphic, which is very good. You should always describe your data in graphics to give to allow your reader to uh, recognize patterns in the potential patterns in the data. But you should also um, calculate statistics to that describe those patterns, that summarize those patterns. And the correlation coefficient is the statistic that we will be using here. It would make sense in real life. You would also be calculating the correlation coefficient. And how to calculate the correlation coefficient? As you remember, we need basically these three elements of values in the numerator and the denominator. So we need these values and we need these values, right? So we need to first calculate, of course, the means of x and y. And having the means of x and y, we can calculate for each value. Let's say the mean of x turns out to be 21 and the mean of y turns out to be, I don't know, 0.98. We would be calculating the difference between for each value, right? This is a single subject, this row. So for this individual, we would be calculating the difference between the observed value and the mean. So it would be 18 minus 21. And this observed value minus the mean of y, so 1 minus 0.98. And those, we would calculate the product of those differences, right? And we would do the same here, calculate the differences, calculate their product. Calculate the differences, calculate their product. We would have 46 products in the end, and we would sum up those products. And then for the denominator part, here, this is simple. For now, we don't have to care about individual observations. We would just calculate, we have the means of x and y. We would calculate the difference between each x value and the mean, and square those differences and sum them up, and likewise calculate the difference between each y value and the mean, square the differences and sum them up, and then take the square root and then perform the division, and there you get your R. Okay, so yeah, make sure you understand how these three components are calculated. Okay, this is simple. The sum of squares you already know. This is slightly more complicated perhaps, but I hope you understand. Again, what we do is, sorry, I want to use the, what we do is subtract the mean from each value and then calculate the product. And then sum those products, calculate the product for the 46 values, sum those products, and that's the value here. Okay, turns out to be 1.1. So because it's a positive value, we can already see the correlation is positive, right? Because the as you can, I hope, readily see, there is no way this denominator can be a negative value, right? It can never be a negative value. So the numerator can be negative or positive. And So the numerator will be determining whether r itself is negative or positive. Okay, the numerator itself. And so given that here the numerator, the sum of products, was positive, the r we will calculate from these values will also be positive. And this is not surprising because already by I we kind of saw a positive looking relationship. There was a deficiency here and also, well, slightly less here. So not surprising that this is a positive value. And here we have these two values, the sum of squares for x and y. The variance, as you see, the variance in x is very large. I mean, the scale is very large. The scale for the y values is pretty narrow, and the scales, of course, here of x and y, they will be influencing also the sum of products, right? But when we divide by these two sum of squares, when we divide the sum of products by these two sum of squares, we get rid of that scale effect, or we scale the numerator so that the relationship is constrained within -1 and 1. Okay, so things get cancelled out basically. And we end up with, in this case, we end up with a correlation coefficient of close to 0.3. Okay, not bad. So this tells us that there is some relationship. In fact, we will we'll learn about this next week more, but you can also, people also use the squared of the correlation coefficient, r squared, as a measure of how much variance one variable explains in another. We learned about big R squared before in ANOVA context. Again, it was how much the group effect, well, it was a ratio of the sum of squares, and it was telling us how much one categorical variable, like groups, is explaining total variance. And r squared, so the squared value of r, in this case, it would be, in our example, it would be something like 0.01, 09, sorry, 0.09. This will also be some kind of measure of how much variance is explained by one variable in the other. Okay, so this is a measure of relationship, but it doesn't tell us just this point estimate itself doesn't tell us how reproducibly different is this value from 0. It is obviously non-zero, but how much does the variance in the data support the idea that if we went out and took another sample, it would also be positive and it doesn't have to be have the same value, but how much can we trust a positive relationship? And we can address this question of how much can we trust the correlation coefficient, how much can we trust the relationship, in two ways. We can calculate a confidence interval for R, or we can perform directly hypothesis testing. And the confidence interval for R, it's slightly new. It involves a log transformation of not R itself, but 1 plus and 1 minus R. So the ratio of 1 plus R and 1 minus R logged times 0.5. We first calculate this Z, this statistic called Z. Its value depends on R, as you see, and this value is supposed to, through this transformation, it's supposed to follow a normal distribution. Uh, and there is also, people have come up with an estimate for the standard error of this value, and that's relatively simple, 1 divided by n minus 3 square root. Okay, in our previous case, n was 46, so this would be 43, I guess. And that will be an estimate of the standard error of z. And then using the this z estimate and the standard error, and the fact that theoretically this value is supposed to follow a normal distribution, we can use the standard normal critical values to construct a confidence interval, a 95% confidence interval for zeta. So zeta here is the population quantity for z. So z is what we calculate from the sample. It's based on the R values. Zeta would be the population correlation, a transformed version of the population correlation coefficient that we don't know. So first we will calculate a confidence interval for zeta, and then we will back transform it into the R scale. Okay, so we said we can use R to calculate the estimate, the Z estimate, and using the sample size, we can calculate an estimate for the standard error. We don't learn the theory behind it again, and we already know this value. This is from the standard normal distribution, 0.05 two-sided, so that value is 1.96. You already know this. And basically, so if we, of course, we know n. If we can also calculate an R, we can plug the R values in, we can get a Z, and we can calculate this 95% confidence interval for the Z. That's what we've done here. Z is calculated using this formula. We know R, we just punch it in, and Z ends up having this value. The standard of Z has this value. We know the critical value from the normal distribution, and that gives us a confidence interval for zeta. Okay, and we can back transform zeta into, we can back transform these values, these lower and upper limits, to the original scale of R. So these are limits for based on Z, right? So we can input this value. First, replace Z with this value, and that will be estimate of the lower limit of rho, because R, as you remember, the population the population correlation coefficient is called rho. So if we punch in these numbers, calculate the ratio, we get the lower limit for rho. And if we punch in this number for Z in the numerator and denominator, we get this number for rho, 0.54. So what does this mean? This is the range for where we expect the population correlation coefficient should stand. Just go through these once more to make sure that you can follow what Z and zeta are. So Z is a transformed version of R, and zeta is the population value, so the population parameter. And we describe the expected range, 95% confidence interval for zeta using this well, basically using this formula. Okay, this allows us to calculate 95% confidence interval, and we can further use the minimum maximum values of this confidence interval to calculate back the values of R, and based on those values, those minimum maximum values, we can come up with a confidence interval for the population parameter. Right, and why do we do this trick? Well, basically R itself, we already said R itself is distributed between -1 and 1. It is not normally distributed. And so basically what we're doing here is by transforming R values into Z values, we're doing this trick. We are we're converting R values into this Z value, which under null hypothesis of no relationship has a normal distribution, okay? Follows a normal distribution means if there's no real relationship, then Z follows a normal distribution, and thus we can use that behavior to use the known values, the critical values from the normal distribution, standard normal distribution, to calculate a confidence interval. Okay, so that was the confidence interval part. In fact, most times people just use a Pearson correlation test to calculate the significance of the relationship. The correlation test has a hypothesis of no relationship, just like the chi-square test or the Fisher's exact test. The two variables are independent. There's no relationship. It's the null hypothesis, where rho is equal to 0. And the alternative hypothesis, if we're running it one-sided, would be rho is not equal to 0. And the Pearson correlation test is in fact pretty straightforward. It uses the t distribution, and we calculate the t statistic itself to compare with the t distribution using R and the standard error of R. And the standard error of R, so don't confuse, here we saw the standard error of Z, or standard deviation of Z. Here we have standard error of R, and that standard error has this formula. Again, we won't be learning how the formula is derived, but simply it is a function of R and N. The higher the correlation coefficient, the lower will be the standard error. As you see, the lower the correlation coefficient because it's squared, well, the lower the absolute value of the correlation coefficient, the larger will be the standard error. And then the standard error is also inversely related to the sample size, which is not surprising. Okay, so this is the standard error, and this is the formula of the t statistic. On top in the numerator, we have R itself. So stronger the R, larger the R in absolute value, the larger will be the t statistic in absolute value. I guess that's obvious to see. And in the denominator, we have the standard error as usual. As you remember, in the t statistic we were calculating for the one sample t-test, for instance, was the mean difference between the observed and hypothesized mean divided by the standard error. Again, here instead of mean difference, we have R as some kind of measure of effect size. And yeah, this follows a t distribution with degrees of freedom, not n-1, though n-2. N-2. Yeah, so by calculating a t statistic and comparing it with the t distribution, so follows the t distribution means if the null hypothesis is true, if there's no relationship, the t statistics we would be calculating this way would have a distribution, would have probabilities that are described by the t distribution if there's no effect. Okay, the t distribution would be a representative of the null hypothesis. And so by comparing our observed t statistic with the null distribution, you remember the same story. This is our null distribution. And by comparing what we observe, the t statistic with the null, we can calculate a p-value. If it's two-sided, then it goes, of course, two ways. Okay. And the example here from the book is about inbreeding depression in wolves. Inbreeding is marrying, well, with relatives, which can happen when population size is small, or it can happen when individuals mate with relatives directly, consanguinity. And inbreeding in general can increase the frequency of, it will increase homozygosity within individuals. It will increase the frequency of homozygous sites, and because some variants are always deleterious, segregating in populations, so, or I should say, recessive deleterious alleles that are polymorphic, that are found at certain frequency in a population, they will tend to cause a decrease in fitness-related traits, depending on the population, depending on the distribution of fitness effects or mutations, etc. Many different effects, but in general, it's known that inbreeding can have negative effects on fitness, and inbreeding depression is this lower fitness, lower overall fitness. It's not psychological depression. And there was the study where they were checking 24 litters. Litter here is family, batch of offspring, okay, batch of offspring, and they've measured how much inbred different groups of offspring are from a single mother, and how related, sorry, they're measuring how related the moms and dads of a group of offspring are. That's the inbreeding coefficient. And they've also measured the number of pups that have survived their first winter. Usually, many mammals they mate during spring or summer, and then they give birth during summer, fall, and then so that the offspring are have reached certain size by winter. But then, of course, first winter is always challenging. And here we have the data, two numeric variables, as you see. So the approach here to use would be correlation. These two variables correlated. And this is how the data looks like. Inbreeding coefficient on the x-axis, which would make sense, is probably explanatory, and number of pups surviving, it's on the y-axis. Lower the inbreeding coefficient, there seems to be higher number of pups surviving. Higher the inbreeding coefficient, there seems to be a lower number of pups surviving. So in general, we see a negative relationship. Okay, and now what we want to do is both to measure the strength of the relationship using what? The correlation coefficient, right? And further to assess if it is significantly different than zero. If we, how much we would expect such a correlation coefficient by chance, given our sample size. Yeah, so how to calculate the correlation coefficient? Again, we would first need to calculate the mean of the two variables, and then calculate the products of the differences for x and y for all the points, right? So it's this part here. For each point, we calculate difference from the x mean and difference from the y mean, the products, and we sum up the products. Overall, we would have 24 such products. We would sum up the products, and the value appears to be minus 2.6. And minus 2.6, that indicates, okay, that we have a negative correlation coefficient. Again, not surprising from based on what we see in the data. And it's always good to double check that whatever you see in a graph and whatever statistics you calculate are in general coherent with each other, consistent with each other. And if you see inconsistency, you can always go back and try to see if there's been some error somewhere, because errors always happen to you, to me, to everyone. So always good to this is also called like sanity check, to make sure that everything in your data and all your statistics make sense. Okay, so this was the first thing we calculated. The sum of products was negative. Okay, makes sense, we said. But then to calculate R itself, we need also the sum of products for, sorry, the sum of squares for x and for y, and basically dividing this value by the square root of these two values, we can get our correlation coefficient, which is minus 0.6, which is, well, pretty, given that the correlation coefficient, the minimum correlation coefficient possible is -1. This is pretty strong. And if we, the R squared that would be calculated from this would be, well, approximately 0.4, as you can see. And that means like 40% of the variance of one variable is explaining, or one variable, the values of one variable are explaining 40% of the variance of the other variable. Okay. So this is pretty strong looking, but of course, to be able to really decide if this is non-random, non-zero or not, we would like to perform perform hypothesis testing, and in real life, everybody would be expecting from you the result of such a formal test, not just present data and showing the R, that R is, has a strong negative value. This is not enough. You would be asked, what is your p-value? Even though, even if it's very obvious to the eye, you would still be asked to calculate a p-value, not to torture you, but it's some kind of standard that allows us to compare results across studies, and that's why p-value is helpful. I mean, yeah, it's a historical thing in a sense. It's not the reporting a p-value is not something that was written in the holy books or something, and it's been around. This type of data presentation and result presentation has only an age of about 50 to 100 years, not more than 100 years, and things might change eventually, but this is the standard now. Okay, so that's what we're learning. We report the p-value and we interpret it, but when interpreting, of course, we're always careful. Just like having the calculating the p-value itself, using a p-value cutoff of less than 0.05, this is just this is totally historical. Why we choose 0.05 but not 0.01, whatever. So one has to be always we have to know that these are just some measurements, some evaluations we apply on the data, and they are not, we are not here proving any relationship or disproving any relationship based on just one sample. And proving, disproving this will happen as more and more data come, as or based on theory. Of course, you can also prove things with theory. So yeah, in our case, what we did, we calculated R, which was pretty negative and strong, we said, and we now need to calculate the standard error, and that's the formula we had learned before. That's correlated with that involves R itself and also the sample size, and the standard turns out to be something like 0.16. The degrees of freedom is 22. The t statistic we calculate from by dividing the R value, the correlation coefficient estimate, and the standard error of the correlation coefficient estimate, and the t statistic turns out to be minus 3.6. And well, we can calculate the p-value for this directly using R, using the pt function. But if we don't have R accessible, then we can just refer to a table of critical values from the t statistic. If we use alpha 0.05 with degrees of freedom 22, as you remember, the degrees of freedom was not n-1, n-2. We have two variables, and the t, the critical value of the t statistic is 2, and given that the absolute value of our t statistic is much larger than the critical value, right? We can comfortably reject the null hypothesis. And again, what was null hypothesis? There's no relationship. R is 0. Rho is, or I should say, rho is 0. That's the null hypothesis. So we can reject the null hypothesis in favor of the alternative hypothesis that there is a relationship. So inbreeding may reduce survival. Again, this is only a sample. We've not, it's possible that this could have been a false positive. We always have to be careful. So being modest about our results, being careful, being cautious is always helpful. Right. Now, we also have to touch upon the assumptions that we were using. So the theoretical work that developed these approaches that figured out that the standard error of the correlation coefficient can be estimated using this approach, and this has a t distribution, etc. So this body of work it has been developed based on certain assumptions about data behavior. One is, of course, that this is a random sample. Observations are independent. They are not replicates. They're not more related to, some observations are not more related to others than average. But we also have this assumption about the distribution shapes. We have two variables, and the assumption is that they have a bivariate normal distribution. So the bivariate normal is describes the shape of the joint distribution of two numeric variables. So both of them have a normal-like distribution. Both of them are symmetric. As you see, x is symmetric, y is also symmetric, and their joint distribution is also symmetric. Their joint distribution would not necessarily have to be symmetric. I think we will see some examples like that. But yeah, if this assumption holds, if they're both bivariate, they have a bivariate normal joint distribution, then we are fine. Yeah, and this is a description of the bivariate normal, that x and y are independently normal. Their relationship is linear, like here, and the cloud of points have a circular or elliptic shape. Ah, yeah, okay. Here we have another example of x and y. They have each normal distributions, and then just projecting the points on one axis or the other axis, we see a normal shape, and then their joint distribution is also elliptic, as you see here, and it has yeah, it is elliptic and not bimodal, for instance. It's bivariate. Don't confuse bivariate with bimodal. Bimodal means with two modes. If there had been, for instance, if there had been two peaks here and an absence of data points in between, this would not have been a bivariate normal distribution. Yeah, some examples of bivariate. So here, for instance, x and y, they are not related at all. If we looked at the distribution from the top, would look like this. Here, they are negatively correlated. And here, they're positively correlated, and so on. Yeah. So how might, in what different ways might two variables, the joint distribution of two variables might might deviate from the bivariate normal? You could have weird situations. You could have funnel-shaped distributions like this. So here what's happening is that with the values of x, as values of x, so x and y are related, but not in their means, but the values of y show increasing variance as values of x increase, as you see. You can have outliers, of course, or you could have non-linear relationships. And these type of relationships would all be deviations from the bivariate normal. And in these cases, then the Pearson correlation test, the p-value we would be calculating using the formulas here, they would not apply, okay? If the assumptions are violated, the p-value is not reliable. So we could try to transform the data. Sometimes transforming, like log transforming, might help. Or we can use a Spearman, we can use a non-parametric correlation test. Spearman correlation is the most commonly used. We'll learn it in a second. It's correlation based on ranks. It's non-parametric, again, just like Man-Whitney U. We could use computer simulations, a permutation test, or we can use some regression that allows for non-linear relationships. Okay, and Spearman rank correlation is our, I think, one before last topic. This is an approach where we rank both data. So we have x and y, two variables, right? We rank both of them, both x and y, and then the values of x and y are ranked independently. And then what we do is we calculate the correlation coefficients. We apply the same procedure, but using ranks instead of the original data. The null hypothesis, again, is that rho is 0. Here, rho s indicates it's the Spearman correlation coefficient. And the p-value we can calculate if the sample size is large than 100, we can use the t approximation just like we learned. When the sample size is small, we use we calculate the correlation coefficient using the approach we just learned, but then to evaluate the p-value, to get an estimate of the p-value to evaluate significance, we use a table called the Spearman table that tells us, given a certain sample size, what's the probability of observing a certain raw value by chance. Yeah, so this has been calculated in Spearman tables. Of course, the Spearman correlation test also assumes a random sample, but it has no assumption about data distributions. It doesn't assume that the X and Y are normally distributed or bivariate normal. Okay, and the book has this weird example about some trick from early, I guess, late 19th, early 20th century, and people recounting, so writing about the watching the trick and how many years have passed since they recounted the having seen the trick, and how impressive they describe what they've seen. And as you see, the more time has passed, it looks as if the more exaggerated the story becomes. I don't know why they use this example, but anyway, so the data, as you see, it's not super nicely distributed. Well, the some of the distributions we saw earlier were also not super nice. Well, yeah, this is also not. I mean, we could have also applied the Spearman correlation coefficient here. But anyway, this is our data, and we want to measure. We want to measure the relationship between the two effects. So years elapsed, as you see here, and the impressiveness score. Years elapsed impressiveness score, and instead of, in this case, instead of using the values directly, we rank them. We rank the two data sets separately. As you remember, please don't confuse, in the Mann-Whitney test, which is a two sample test, in the Mann-Whitney test, we had two groups, same numeric variable observed in two groups, and when we ranked them, we pooled the data. We ranked the data and then separated the two groups. And so we had the rank one, the sum of ranks of one group, and the sum of ranks for the other group, right? This was for the two sample case. Here we're doing something different. We have two different variables. Okay, so we rank both variables within itself. Here, for instance, the first one gets 1, and then 2, and then 3 and 4. 3 and 4, we assign them half the average value, and so on. And for the impressiveness score, the same. There are limited number of scores. And okay, then what we do is basic same thing. We calculate the mean of x, and we calculate mean of y, and we calculate for each value the difference between that value and the mean of x, and for each y value the mean of y, and we calculate the products of these and the sum of products, and that's this 566. Okay, here are, I think, was the years, yeah, the year values and the impressiveness score values, the sum of products, and the sum of squares for years, and for impressiveness score we calculate the the ratio, of course, after calculating the square roots of the sum of squares. Here we get a correlation coefficient of 0.78, Spearman rank correlation coefficient. Okay, we did the same thing based on ranks. That's basically it. And yeah, so for in the Spearman case, people have calculated theoretically how R values would be distributed if there's no real effect. Under the null hypothesis of no real effect, how R values would be distributed under different sample sizes. Okay, and for instance, it seems that when sample size is small, when it's only 5, there is a 0.05% chance of observing an R value of 0.9 or larger. Okay, so this can happen by chance, 1 in 20 chance when sample size is small. But yeah, it would be happening relatively rarely, even by chance. So if we reach this R value, we can say, okay, our P value is at 0.05. But such high R value will not be expected if the, as you see, if the sample size is higher and higher, right? I'm actually slightly surprised. There might be a mistake here because the values should be monotonically going down. There's no reason for them, for this value to be higher. I think there must be a mistake here. There must be a mistake. Yes. I'll double check. But yeah, we can also use R to, in fact, calculate what the R values would be, the critical values corresponding to these alpha values. If you can do that yourself, if you try to do that, this would be super fine. Okay, this is basically how we use that table. So these are the critical values for R. If the observed R is larger than for a certain sample size, this is not degrees of freedom, it's directly sample size. For a certain sample size, if degrees of, for a certain sample size, R is beyond what we observe, positive or negative, doesn't matter. In this is two-sided. Then we can conclude that alpha, the p-value must be lower than this alpha value. So in our case, we had a sample size of 21, right? 21 observations, yes. And we calculate an R value of 0.78. So at 21.78, it's beyond this critical value. For alpha 0.05, it's also beyond this critical value. So we can comfortably say the p-value must be less than 0.01. We could, of course, also calculate it directly. So we can reject the null hypothesis in favor of a relationship. And if the sample size is large enough, then we can calculate the p-value using the t distribution. Okay, it works the same way. Okay, and a couple of final points about correlation. So, and the correlation coefficient. So the correlation coefficient depends on noise. I guess you've already got an intuition about this. So if the data is more noisy, a relationship between two variables will appear weaker, right? Why is that? Well, basically, let's go back to the first formula. The more the noise, the higher will be the the denominator here, the weaker will be R, right? Just like here, compare this case with this case. They're both positively correlated, but more noise in X and Y here, causing a lower R value compared to here. Among the total variance, there's less explained. Sorry. R can also be influenced by data range. So if we are studying the relationship between two variables, X and Y, if we study the wider the range of values we study, the more, the easier, if there's a linear relationship between them, the easier will be to observe that linear relationship. The narrower the range, the less we can observe. That will be clear in a second with an example. And also, sample size influences variation in R, such that if sample size is small, we can get large R just by chance. And okay, first think about how noise affects R values. Well, yeah, if we add more noise, you can do this with an experiment, small simulation experiment in R as well. When if you know how to randomly collect data from a normal distribution, you can simulate such data with high standard deviation, low standard deviation, and see that with more standard deviation, a relationship will become dilute and more dilute, and weaker R values. With respect to the range, so here we have two variables that are negatively related. Obviously, across the whole range, the correlation coefficient is minus 0.77. But if we just look at a sub part of the data, okay, a narrow range of the data for X, then if you would you also guess that the relationship, the correlation coefficient we would calculate from just this part of the data would be as strong? No, it would be much weaker, right? So the power of the correlation coefficient, it depends on the range of the data. The wider the range, if there's a linear relationship, the wider the range, the stronger the easier we can see a relationship. And also, yeah, finally, the smaller the sample size, there will be more noise in R, and high values can appear more easily. So here, I did a small simulation experiment myself. I collected data, random data with no relationship between two groups, here shown with N1 and N2. So these are the two groups, 1 and 2, and their sample sizes were 5 in both cases, and I calculated correlation coefficient between them. And frequently, as you see, we get relatively high or negatively high positive or high negative correlation coefficients. Whereas if I do the same experiment when sample size is 25, then such large positive or large negative correlation coefficients, they never occur, right? And that should be also obvious to you. Okay, and last point. In some experiments, we have you can have multiple X and Y values. In that case, let's take not this example, but let's take this example. Okay, let's say we assuming wolves are wolf pairs are monogamous. I think they're not, but assuming they're monogamous, the same pair mates the next year, and we can observe the same inbreeding coefficient. Thus, we would be able to observe a second set of values. Could we just pool all the data together and calculate the correlation coefficient and the p-value? How would the correlation coefficient be affected? How would the p-value be affected? if we have X or Y values or both, uh, how to say, um, collected information for X and Y values collected multiple times, how will this change R and how will it change the p-value? And again, you can already guess how things would work. You can calculate it by a formula, but a simple way to study how data behaves is just running some small experiments in R. So I generate two random data sets of size n using this R norm function, collecting data, randomly collected data from a standard normal distribution, and I calculate a correlation between them. And the correlation coefficient appears to be minus 0.225. This is the data. And does there seem to be a relationship? Well, not really. And by chance, we got such a correlation coefficient, but probably not significant. But how about with the same data, I just repeat X values and Y values 20 times. The same values 20 times. The correlation coefficient, as you see, stays the same. Would the p-value stay the same? No. Here the p-value was not significant. Well, it was random data anyway. They weren't related, so we don't expect that this minus correlation appear just by chance, and we couldn't reject that the true correlation is not equal to 0, right? P-value is pretty high. But if we have now 20 times more data, as you see, degrees of freedom 8, degrees of freedom 198, then we can comfortably reject the null hypothesis, right? But the same relationship. So larger the sample size, we can identify small effects. But in fact, here we didn't have really independent data. So this would have been a case of pseudo-replication. So the p-value would be unreliable because data points aren't independent. What we could do, well, we could use a very simple approach. If we have multiple data points, we can simply, as in this case, like for instance, in this problem, if you had another set of values here from, if this was year 1 and this was year 2, okay, imagine this was 5, this was 4, I don't know, blah blah. If you had such second set of values, you can pull them all together because these belong to the same family, right? Same family. But what you could do, you could calculate the average of these values. That would be the simplest and safe approach. Calculating the mean, and then each data point then is independent, and you then calculate the correlation. You could also use some special formula that takes into account this effect. And yeah, you can also go for some regression models, but the simplest solution might be just taking the average. Okay, so please solve the assignments, all the problems at the end of the chapter, and we see each other on Monday. Thank you.